Then we can go through the chemical mutagens and chemical mutagenesis. Muller's discovery of the mutagenic effects of radiation provided a method for the induction of large numbers of mutations with a minimal amount of effort. But these studies on radiation-induced mutation provide little information about the molecular details of mutation processes. Subsequent discoveries of the mutagenic effect of a large variety of chemicals, many with very specific effects on DNA, provided the tools needed to work out many of the details of mutation processes at the molecular level. About chemical mutagenesis and chemical mutagens. Chemicals are introduced in the environment as part of industrial activities. These chemicals are known to have slight to very strong mutagenic effects. The permanent heritable mutations occurring in organisms due to chemical mutagens or mutagenic agents are called chemical mutagenesis. The chemical mutagen is very stage specific. Accordingly, chemical mutagens can be classified into two classes: chemicals which are mutagenic to both replicating and non-replicating DNA, for example, alkylating agents and deaminating agents, and chemicals which are mutagenic only to replicating DNA, for example, base analogues and intercalating agents. It has long been recognized that most of the strong mutagenic agents are also carcinogenic agents because most of the genetics agree that somatic mutation can cause cancer. There are a number of chemicals which can act as a potential mutagenic agent in humans, and some of these chemicals are also used as drugs for curing some diseases. And the first chemical mutagen discovered was mustard gas during World War II by C. Aberrations and her associates. This is the structure of the sulfur mustard. The four major classes of reactive chemical agents that can cause mutation are deaminating agents, and these deaminating agents are direct acting chemicals. They are directly acting on the base structure of DNA, particularly nitrous acid or HNO2, or compounds that can be metabolized to nitrous acid or nitrates. And second class includes alkylating agents and hydroxylating agents. These are mutagenic to both replicating and non-replicating DNA. Alkylating agents cause alkylation consisting of a small alkyl group such as methyl or ethyl group to nitrogen atom or oxygen atom in one of the bases in DNA. For example, mustard gas. And these are the chemical structure, and we can see the four different groups of chemical mutagens. The base analogues, which include 5-bromo-uracil, the analog of the base uracil, then aminopurine, that is an analog of adenine. Then comes acridines. They are examples of intercalating agents. For example, proflavin, that is 2,8-diaminoacridine. Then comes third group, alkylating agents. For example, sulfur mustard, that is di-2-chloroethyl sulfide. Then nitrogen mustard, di-2-chloroethyl methylamine. Then ethyl methane sulfonate or EMS. Then comes the fourth group of chemical mutagens, deaminating agents, which include nitrous acid. Then one miscellaneous group is also there, which is usually included along with alkylating agents, that is hydroxylating agents. For example, hydroxylamine NH2OH. Hydroxylating agents usually hydroxylates the amino group of bases like cytosine. And the base analogues I have told you, the acridines, the base analogues which include the 2-aminopurine and 5-bromo-uracil are compounds that can stimulate or mimic the normal bases present in DNA and may be incorporated into the DNA chains in place of normal bases of DNA. And intercalating agents, they are usually flat 3-ringed aromatic molecules with approximate dimensions of a base pair, and therefore they can intercalate or slide in between two adjacent base pairs during replication and increase the probability of mistakes along the double helix. For example, the acridine. Next, we can see the details of the mutagenic action of deaminating agents. Here, the mutagenic action of nitrous acid is given, and this nitrous acid, which is formed from organic precursors such as nitrosamines and from nitrate salts, is a very potent reagent capable of removing the amino groups from the bases like cytosine, adenine and guanine. Usually, these three bases contains the amino group in their structure. Nitrates and nitrites are used as preservatives in sandwich meat and other meat products. Nitrous acid greatly accelerates the oxidative deamination of cytosine to uracil. Occasionally, deamination occurs spontaneously, so that some spontaneous mutations are probably due to this process. And therefore, we can say the spontaneous mutations can also lead to deamination. And this is a structural change occurring in cytosine after deamination, and we are getting the structure of uracil after the removal of the amino group. Uracil formed by deamination of cytosine can pair with adenine. This results in GC to AT transition mutation after the replication. The GC base pair is converted into AT after the replication. Similarly, nitrous acid deaminates adenine to hyposandine and guanine to yield sandine. When this occurs, DNA may not function in the intended manner, causing a mutation to occur. Usually, the mutagenic action of nitrous acid is exerted only through the changes in adenine and cytosine. The hyposandine formed pairs with cytosine in the next replication, and it causes AT to GC transition. If you check the first replication, second replication, etc., you can see the changes coming in the base pair after deamination. Deamination of guanine is not directly mutagenic like that of adenine and cytosine because the sandine formed can again base pair with the cytosine. Therefore, there will not be change in the newly formed base pair. Thus, the mutagenic action of nitrous acid is exerted only through the changes in adenine and cytosine. And here you can see the structural change occurring in adenine after the removal of amino group or after deamination. We are getting the structure of hyposandine, and in the case of guanine, after deamination, we are getting the structure of sandine. And we can see the repair mechanisms as in the case of physical mutagenesis. Here also, the chemical mutagenesis also, we can see some repair mechanism going on in all the organisms. For example, the uracil's hyposandine and sandine residues formed can be recognized and removed by specific enzymes. For the removal of uracil residues, uracil DNA glycosylase enzyme acts first, followed by the sequential action of DNA polymerase I and DNA ligase for the removal of uracil residues by the cleavage of sugar-nitrogen bond of uracil to release the base from the sugar-phosphate backbone, then repaired by the repair system. And here, the uracil DNA glycosylase is also known as UNG or UDG. Its most important function is to prevent mutagenesis by eliminating uracil from DNA molecules by cleaving the N-glycosidic bond and initiating the base excision repair pathway. Therefore, this is usually taking place during the repair mechanism. Next is about the alkylating agents and hydroxylating agents. I have shown you some examples of alkylating agents, and here some other examples are also given: dimethyl sulfate, dimethyl nitrosamine, then ethyl amine sulfonate, and methyl methane sulfonate. And formerly I have given the examples of nitrogen mustard and sulfur mustard. All these are alkylating agents which can cause changes in the DNA bases during replication. And these are the structural representation of dimethyl sulfate, dimethyl nitrosamine, di-2-chloroethyl methylamine, di-2-chloroethyl sulfate. Alkylating agents cause alkylation consisting of a small alkyl group such as methyl or ethyl group to the nitrogen atom or oxygen atom in one of the bases in DNA and alter their base pairing potentials, and it leads to mutation. Therefore, the alkylating agents exhibit less specificity in their mutagenicity. For example, the highly reactive chemical dimethyl sulfate can methylate a guanine residue in the DNA to yield O6-methylguanine or N7-methylguanine, which is unable to base pair with cytosine, the normal partner of guanine. Usually, guanine base pairs with cytosine, but after the conversion or methylation process, the newly formed base O6-methylguanine or N7-methylguanine cannot base pair with the guanine, but they can base pair with cytosine, but they can base pair only with the thymine. Therefore, the normal replication is disturbed and mutation results. Therefore, there is a transition mutation. Studies also show that alkylating agent induce all type of mutations including transitions, transversions, frame shifts, and even chromosome aberrations with various frequencies depending on the specific alkylating agent employed. Repair of alkylated bases. Both bacterial and animal tissues contain enzymes that can specifically remove O-methylguanine or other alkylated bases and replace it with normal base guanine, again by a cut, patch and splice mechanism similar to that in the case of repair of conversion of cytosine to uracil. Therefore, almost all the repair mechanisms are similar, and it includes a DNA polymerase 1 and 2 involvement for the repair. Then comes the hydroxylating agents. An example of an hydroxylating agent is hydroxylamine NH2OH. It has very specific mutagenic effect. It can cause transition mutation in DNA. The exact mechanism of hydroxylamine mutagenesis is still uncertain. It apparently acts by hydroxylating the amino group of cytosine. The resulting hydroxylamine cytosine can base pair with adenine to produce GC-AT transition. And these hydroxylamines, due to their specificity, has been very useful in classifying transition mutation. A single oral or intraperitoneal dose of hydrazine administered to rats, mice, guinea pigs and hamsters in experimental studies resulted in the rapid formation of N7-methylguanine or O6-methylguanine in liver DNA, which was not detected in controls. Therefore, there is a change in the structure of the bases after exposure to hydrazine. And repair of damaged bases, I have told you, usually the repair process occurs with the help of an endonuclease and exonucleic action of DNA polymerase, and then there is a synthesis of new nucleotides and they are getting attached to the strand and then the DNA ligase comes and it causes the ligation and healing of the strand, and we are getting a newly repaired DNA duplex. And the third category of chemical mutagen, base analogues. A base analog is a molecule sufficiently similar to one of the 4 bases that it can be incorporated into a DNA duplex in the course of normal replication. A base analog is a mutagenic chemical which can substitute a normal base and can be incorporated to DNA during replication. And because they are correctly hydrogen bonded to a base on the template strand, they are not excised by proofreading activity of the DNA polymerase. And they are weak mutagens, and in order to produce a reasonable number of mutations, bacteria must be grown in their presence for many generations. Some base analogues are mutagenic because they are more prone to mispairing than normal nucleotides. Therefore, these base analogues usually resembles one of the bases and they can get incorporated into DNA during DNA replication and cause changes in the DNA. 2-aminopurine is a base analog of adenine, and normally in its usual state it base pairs with thymine. Occasionally, in the rare form, due to tautomerism, it can be converted into imino form and can pair with cytosine by the formation of 2 hydrogen bonds. And the mechanism of mutagenesis by 2-aminopurine. When 2-aminopurine is incorporated into DNA in the place of either adenine or guanine, it causes transition mutation. That is, during the second replication, you can see there is a change in the base pair from AT to GC and also GC to AT. Although 2-aminopurine is only rarely incorporated into DNA, once incorporated, it can act as a potential mutagen. And this is about the 5-bromo-uracil. It is a base analog of thymine. The common keto form of 5-bromo-uracil base pairs with adenine, but rare enol form can pair with guanine. 5-bromo-uracil, like 2-aminopurine, induces both AT-GC and GC-AT transitions after replication. As base analogues induces transition in both directions, the mutations induced by a base can also be reverted by a base analog. For example, modern DNA sequencing methods are based on the use of dideoxynucleotide analogues to terminate elongation at predetermined sites. And these dideoxynucleotide analogues are used in the clinical treatment of various diseases including cancer and viral infections like AIDS. And one of the most important drugs in the treatment of AIDS is the dideoxy analog AZT, which slows down replication of the HIV virus because on replication the DNA of the virus incorporates AZT in preference to normal deoxythymidine. And AZT is 3-acido-2-3-deoxythymidine. And next is about the intercalating agents. Examples of intercalating agents are 9-amino acridine. Then another example is ethidium bromide, then proflavin, and some acridine dyes like acridine orange. And these are usually having a structural similarity of base pairs in DNA. These are flat 3-ringed aromatic molecules with the approximate dimensions of a base pair, and they can intercalate or slide in between 2 adjacent base pairs along the double helix during replication. In some unknown way, this causes errors at the next replication. Most frequently, one base pair is added to the DNA sequence, or less frequently, a base pair is deleted. the result is a frame shift mutation. Therefore, either a base pair can be added or a base pair can be deleted according to the concentration of these intercalating agents. And you all know that the most frame shift mutations are lethal in nature. And this is a diagrammatic representation showing the mutagenic action of acridine. Addition and substitution of nucleotide caused by the presence of acridine dyes during DNA replication. You can see a normal DNA strand here, and once in the medium, if there is acridine, they can intercalate in between the base pairs, and during the replication, two types of changes we can expect: either deletion of a base pair or insertion of bases. And therefore, in the daughter DNA, it is reflected. And it was found that when present at relatively low concentration, acridine acts by becoming inserted between adjacent base pairs in the DNA. And if the acridine is inserted into the template strand, and during replication, a randomly chosen base pair is inserted opposite the acridine molecule when the replication fork passes by. At the next round of replication, the correct complementary base is paired with inserted base, with the result that one base pair is added to the DNA in that region. And this is called insertion mutation. And in certain cases, if there is high concentration of acridine, the acridine becomes inserted in the newly synthesized strand. It blocks one of the bases of the template strand from having a complementary base during the next replication. And then if the acridine is lost before the next round of replication, the result will be deletion of a base pair. Then also there will be a shift in the frame of the DNA strand, and therefore we call it as a frame shift mutation. Therefore, we have gone through the action of different types of chemical mutagens, and in summary we can say the mode of action of various chemical mutagens will give different types of base pair changes. For example, the first case, in the case of deaminating agents, here it is given as nitrous acid. We are getting a two-way transition mutation, that is the AT base pair in the parent strand is converted into a GC base pair in the second replication, and therefore we can say there is a AT-GC two-way transition. Then in the second case, in the case of hydroxylating agent, for example hydroxylamine, a GC-AT one-way transition occurs. And in the third case, in the case of alkylating agents, a AT-GC or GC-AT two-way transition occurs. And in the case of base analogs, for example fibromo-uracil, AT to GC, a two-way transition occurs after the second replication. And if there is 2-aminopurine, then also AT-GC two-way transition occurs. And in the case of intercalating agents like acridine, there will be a frame shift mutation. Either an insertion of a base pair or deletion of base pair occurs. Many other chemicals appear to cause mutations, and public awareness of this is increasing as industrial effluents, cosmetic ingredients, food additives, etc. are examined carefully for any mutagenic activity in test organisms. Next is about the mutation rate or mutation frequency. The mutation rate is the probability that a gene undergoes mutation in a single generation or in forming a single gamete. And the mutation rate is generally defined as the average number of mutations per cell per division, or the frequency with which the genes mutate spontaneously. The mutation occurs much more frequently in certain region of the gene. and the measurement of mutation rates is important in population genetics, in studies of evolution, and in analysis of the effect of environmental mutagens. What is the biochemistry of mutagenesis? The order in which bases occur in DNA determines the nature and structure of newly produced RNA, which is generated during the synthesis of new protein and enzymes in cells. Exchange, addition or deletion of any nitrogenous bases in DNA alter the nature of RNA produced and can change vital life processes such as synthesis of an important enzyme. This phenomenon, which can be caused by symbiotic compounds, is a mutation that can be passed on to progeny, usually with detrimental results. Therefore, if there is a change in the bases in DNA, it will alter the nature of the RNA produced and it will result in the synthesis of a peptide which is having a different amino acid, and it will cause a change in the function of the protein formed. Therefore, mutation of some 2500 different human genes have been detected to date. Many impair a function or ultimately cause death of the organism. That is, it is lethal. The relationship of mutation rate to cell number is given in the following equation. Mutation rate is equal to 0.69 mu by n minus n0, where mu is the average number of mutations occurring when n0 cells increase in number to n cells. The mutation rate of a given culture can be determined by growing a population of cells in a solid medium where each mutation gives rise to a mutant clone that can be detected as a single colony. Mutation rate per gene in bacteria is in the order of 1 in 10 lakh in 10 million per cell generation. Each gene had its own characteristic mutational behavior. Some genes undergo mutations more frequently than others in the same organism. And these are the references. Principles of Genetics, 4th edition by Gardner, then the Principles of Genetics by Strickberger, then Understanding Genetics and Evolution by Tyagi R. You can go through these references also. And next is about, usually we are giving some topics for writing short notes in record book. And here I am giving some topics, and you have to write some short notes with diagrams, and you can send it to me through mail. And first is the topic on giant chromosomes, then second one prokaryotic and eukaryotic ribosomes, its composition and function, and third PCR and its applications, and fourth one plasmids, types and features, and fifth one Human Genome Project. Sixth one, diseases caused by numerical aberrations of chromosomes. Then seventh one, mutation detection methods in genetic disorders. And then eighth one, beneficial mutations and practical applications of mutations. And ninth one, about environmental teratogens. And 10th one, human genetic disorders. Therefore, you have to write all these and send me or submit in the Google Classroom.